Hi, welcome to Enzymes with Eduardo. Today we'll be talking about the hydrofolate reductase. The systematic name of this enzyme is EC1513. The first number 1 indicates that the reaction is oxidoreductase. The number 5 tells us that the bond where it reacts is in the CH-NH bond. The second number 1 let us know that the molecule NAD or NADP is involved in the reaction. And finally, the number 3 indicates that the reaction happens in the hydrofolate reductase enzyme. This enzyme is involved mainly in 3 metabolisms in nature. First, in the tetrahydrofolate metabolism, mostly called the vitamin B9. It is also involved in the transformation of folate 2 for the plants and folate 3 for the E. coli. Focusing on the first metabolism, the tetrahydrofolate metabolism, the hydrofolate is in charge of oxidizing the NADPH to obtain the reduction of the folate, producing the hydrofolate and NADP. This cycle is repeated until it's formed the tetrahydrofolate. When this enzyme has a failure in its function or there is just a lack of it, it can provoke a disease called megaloblastic anemia, which is caused when there is no folic acid or vitamin B9, because it wasn't produced by the reaction of this enzyme. The folic acid has important functions during the synthesis of DNA to codify specific proteins. In this manner, when there is a lack of folate, it can cause intractable seizures and mental deficits. It is important to understand the thermodynamic side of these reactions. For this kind of enzymes reaction, the spontaneity can be obtained by clearing the Gibbs free energy from the equilibrium constant equation, and by taking as a reference the ideal gas constant and a value of the equilibrium constant obtained of literature. We can calculate this value the following way. As we can see, the value of the Gibbs free energy is negative, indicating that this is a spontaneous reaction, also called exorganic process. Talking about the maximum velocity of this reaction, we can obtain it from the relation enzyme-substrate based in the Michaelis-Menten equation. Using the dissociation constant and the substrate concentration, we can know that the maximum velocity of this enzyme reaction is 1.75 units over milligram. On the other hand, and based on the database Brenda, we can know that the Michaelis constant for humans is 0.0019 for dihydrofolate and 0.0074 for NADPH. This indicates that the NADPH molecule has more or less affinity than the hydrofolate for the substrate. This data helps us know that the relationship between the substrate and the inhibitors involved. In this manner, we can know how to attack the disease induced by the failure of this enzyme. This enzyme has inhibitors that are synthesized naturally by the human body. However, there are others used to treat specific diseases. Firstly, we have methotrexate, which has a competitive inhibition of the folic acid. This one has been used to treat cancer by finishing the tetrahydrofolate so the cellular proliferation stops. The inhibition constant of this inhibitor was obtained from research in human placenta, and it has a value of 6.1 times 10 elevated to 12. Another example of inhibitor is the pyrimethamine, which this one also has a competitive inhibition, and it stops the sequence steps for folic acid production. And the constant inhibitor for this inhibitor has a constant of 2.2 times 10 elevated to 9. Other examples of inhibitors of this enzyme are the aminopterin and the triamterene. A proposal to treat the disease caused by the malfunction of the enzyme of interest is using therapeutic recombinant proteins. To obtain this biotherapeutic, we have to treat the restriction enzyme in a vector of clonation in the DNA that contains the gene that codifies the protein of interest. And well, in this explanation, a vector is a DNA molecule that acts as a clonation transport. An example of these molecules are the plasmids. Next, we have to join the gene of the protein with the vector to form a DNA R. Then we have to multiply or clone this DNA are produced in a host cell, such as E. coli. This way the gene has to be expressed in the protein. Regards the production of treatments like the one explained previously and how they impact on society, we can tell that they are much more efficient and cheaper than regular drugs. This means that the impact is not only on the health sector, but also in the economic one. By this, we can tell that the third world countries can have more opportunities to be involved in these treatments, because these methods are not as aggressive as others. In these manners, some of the Sustainable Development Goals of the UN attacked by this biotherapeutic are the third one and the ninth one. The number 3 talks about health and well-being, and the number 9 talks about the innovation of industries. In these kind of methods, they keep coming up as investigation continues. Personally, I think that all this information needs to be known by everybody, so it can have a support that it deserves. I think that it's amazing how the modifications that are made in a micro level have very important effects in a macro level. It doesn't matter if these changes are made by humans or because of malfunction in the DNA. Also, something I find very interesting is the continuous investigation of the metabolic pathways. They are much more complicated than I expected. However, when I started to comprehend a little bit more about them, I knew that they led up to much more complicated concepts. And this just made me more interested about these topics. This made me realize also that the effects that these pathways have in our bodies and how we can change them or maybe modify them so we can obtain different products of them.